Adaptive immunity. So, adaptive immunity is one branch of the immune system, and it really refers to the branch of the immune system that recognizes specific antigens, or actually specific areas of antigens that are called epitopes. And an epitope is just a section of an antigen that can be recognized by an antibody. Okay, so it's recognized by an antibody, and this antibody could be floating freely in the bloodstream in the form of immunoglobulins, or it could be attached to a cell, a lymph cell to be precise. So, and the cell could be either a B lymphocyte or a T lymphocyte. Okay, so the adaptive immune system is the part of our immune system that responds to specific antigens by binding with antibodies. And why is it called adaptive? Well, it's called adaptive because when we are born, we actually have, our body produces, millions and millions of T cells and B cells that have very specific antibodies attached to them that can only respond. Each one of these can only respond to one antigen, right? So, okay, like that. So each one of these, and there are literally millions of these, and what happens is, if, most of these are never activated in our lifetime, right? And how does our body develop all of these? Well, we understand part of the process, but it's an extremely complex process of how DNA is sort of broken into pieces to produce these different protein forms. Um, and so we literally have millions of these, and they are precisely, each cell can only respond to a epitope with a very specific shape, right? So this one can respond to things that are shaped like that, and this one here can only respond to things that are shaped like this, and this one here can only respond to things shaped like this, and this one here can only respond to things shaped like this. Okay, so what happens is, we've got millions and millions of these stored in our lymph nodes around our body that are developed in our bone marrow and our thymus tissue at a very early age. And, we may only have a couple thousands of each type of cell, and they're really not enough to fight off an infection. But what happens is, maybe some of these are B cells and some of these are T cells. What happens is, if they are activated, then they are going to start the process of dividing, and make exact clones of themselves with this mixed up DNA that is precisely designed to form this immunoglobulin. So if it's a T cell, it's going to split up into a T cell with these exact antibodies on them, right? And it's going to keep dividing and dividing and eventually make millions of them. And then we will have enough to do something. Okay? And this is why it's called the adaptive immune system, because our immune system can adapt to develop T cells and B cells that are specifically targeted to fighting pathogens with very specific epitopes, very specific proteins or peptides with specific areas on them that they can recognize. Okay, so T cells will divide when they get activated by their epitope, and B cells will also divide when they get activated by their epitope. And B cells can actually divide when they divide. They can differentiate themselves into two different types. They can differentiate themselves into memory B cells and make a whole bunch of memory B cells. And these memory B cells will just hang out in the lymph tissue, and they won't be immunologically active, but they will just stay there for a later time. So that way, when instead of there only being 1,000 or so cells with a specific antibody on them, now there will be maybe hundreds of thousands or millions of cells. So the next time the body sees this specific pathogen with this specific protein, it will respond much, much quicker because you're starting out with a much bigger base of cells that have this antibody. Okay, so those are memory B cells, and then the other cell that B cells differentiate to when they get activated are plasma cells. And plasma cells are cells that have the machinery to generate lots and lots of antibodies, which are designed not to stay on the cell membrane but to be released into the bloodstream, and they become immunoglobulins that are, again, specific. They're exactly the same, made by the same DNA as the ones that recognize this epitope, and so they will move into the bloodstream to defend against the pathogen that has this protein on it, or maybe bacteria or virus, or just a protein that is in the bloodstream, perhaps a toxin, to neutralize it. Okay, so that was just a very quick introduction to the adaptive immune system. Now I want to talk a little bit about some regulatory aspects of it. Now, if these T cells and B cells just reacted to every single possible protein that had this shape inside the body, then we could have a problem, because a lot of proteins have similar shapes, and we may have proteins on our own cells that have those shapes. So the T cells, and we don't want the T cells and B cells to be unregulated and to attack proteins that aren't necessarily part of a pathogen or a threat to the body. So one of the ways that we regulate this is through what's called the major histocompatibility complex, which is in the human body synonymous with the human leukocyte antigens. Okay, so this system essentially works by preventing the T cells and B cells to react to an epitope unless it is presented with a human leukocyte antigen as well. So this requires the activity of macrophages. So, and this is how one of the ways that the innate immune system and the adaptive immune system sort of work together. So let's say we've got this little bacteria here, and it's got these proteins on the cell wall in a nice triangular shape, and we've got a B cell over here, and it just happens to actually, the B cell would be much, much bigger than a bacteria, but let me draw that bigger. And it happens to have, I'll make this the nucleus, it happens to have an antibody that matches perfectly with this antigen. But this binding here will not stimulate the B cell. In order for this B cell to be stimulated, what has to occur is first a phagocytic cell has to come and engulf this bacteria and break up its parts, break it up into little pieces. And then what it does is it presents this antigen on a major histocompatibility complex protein, or adjacent to a major histocompatibility complex protein. And then, I'll have it travel over here, and actually it travels into a lymph node. And a lymph node, it actually travels into the lymph tissue. So, travels into a lymph vessel, which opens up into a lymph tissue. And lymph tissue is actually, just has all these little tiny pathways that meander through and past a whole bunch of white blood cells. And actually, let me make that all bigger. So it is sort of this place for white blood cells and macrophages to meet. Okay, so we're going to make a whole bunch of white blood cells here. And each, there's literally millions and millions of white blood cells inside these, and they're lining the walls of these little lymph vessels inside the lymph nodes. And each one of these is going to have a different type of antibody. And as the as the macrophage kind of winds its way through with displaying a major histocompatibility complex, and next to it the little antigen, it's going to connect with the major histocompatibility complex and also connect with the epitope that matches its antibody. And it's only the combination of this connection that will activate the B cell and activate it to divide itself into many B cells, memory B cells and plasma cells, right? And the exact same process occurs with T cells as well. Okay, so the B cells and T cells cannot be activated by an antigen unless that antigen is presented by a phagocytic cell with the major histocompatibility complex next to it. Okay, and this kind of shows, since this lymph node system is really what allows our body to respond to antigens that are in the body, even though we only have maybe a few thousand with each type of antibody. They will eventually get exposed to a new antigen because the body has the system of filtering everything through lymph nodes that are taken up by macrophages. So that just sort of increases the likelihood that they will meet up with one of these. So it's not really a random process, it's a very organized process that increases the likelihood of a B cell specific for an antigen to meet up with that antigen if it has entered the body. Okay, so let's talk specifically about B cells, and we'll review some of the process, and then we'll begin talking about some more specific elements. So again, we've got a big giant B cell here, and it's got this specific antibody, and that's the only antibody that it has. Each B cell or T cell only has one antibody that can recognize one type of antigen, right? And beside each of these, it has a major histocompatibility, or HLA, human leukocyte antigen, that is specific for our body. Okay, so how does a B cell get activated? Well, the first step is a macrophage that has engulfed a bacteria and broken it up into its little pieces parts. Will come up to the B cell with a protein that matches the major histocompatibility. So these two proteins bind with each other, and also presenting right next to the major histocompatibility protein presents the antigen. And now this is called antigen presentation. That's the first step. Antigen presentation. Now the next step is going to be cell division. So we're talking about B cells here. Now again, there's two pathways of differentiation now. The B cell can differentiate into a plasma cell, and the plasma cell is going to start cranking out lots and lots of these immunoglobulins into the bloodstream that are specific to that antigen. Okay, and then the other thing that it can create is it can differentiate into is a memory B cell. And memory B cells are just going to go hang out in the lymph nodes and wait for a second infection. Okay, so step 2 is differentiation. And division. So we're increasing the number of plasma cells and memory B cells specific for this infection. So what happens next? Well, if we have a bacterial infection, the bacteria, that are floating around the bloodstream are going to be attacked by the immunoglobulins. So the immunoglobulins are just going to bind with these proteins, and they're going to have various effects. One of them is called agglutination, and that just means they're sort of combining all of the pathogens or toxins together into a big sort of conglomerate, so it inactivates them. Now, if we are just dealing with a toxin, so say these are toxins that are being released into the bloodstream, one of the advantages of agglutination is you end up combining a whole bunch of particles together in this big globular mass, because these all sort of stick to, it's making everything sort of stick together, and it causes it to precipitate. So it's no longer soluble in the bloodstream, and this precipitate will just get excreted. So it's going to inactivate and excrete. So it precipitates toxins. And then also, remember, one of the effects is it activates complement, and causes inflammation. and it actually can also be directly destructive to the bacteria as well. So bactericidal. Okay, so let's talk a little bit about the memory B cells. So the memory B cells, the B cells get activated and they differentiate into plasma cells and memory B cells, and then the memory B cells just sort of go off to the to a lymph node somewhere and just happily live there within the lymph vessels, just awaiting. Now, what's the advantage of that? I'm sure most people remember a chart like this where we talked about primary and secondary responses to infections. Now, remember, typically our bodies do not have a lot of immunoglobulin. This is maybe 10 to the sixth antigens per milliliter, maybe 10 to the third, and 10 and 0. We have a little log scale here. So if we've never been exposed to an antigen here before, let's say it's varicella, chickenpox. In the beginning, we've never been exposed, so we're going to have a plasma level of near 0. It's not exactly 0 because we do have B cells that sort of randomly produce plasma cells even without exposure. So we'll have low levels of pretty much all of our immunoglobulins even if we haven't been exposed to something before. Now, when we have an infection, when we're exposed to the varicella, it takes our body a little while. So let's say we have a primary exposure here to varicella. We take our 5-year-old daughter over to a friend's house and they have chickenpox. This is in the days before we had vaccines. And we're going to have a delayed response as the body is it takes a little while to recognize for that antigen to come across a B cell with the correct antibody, and then that B cell is starting to proliferate into memory cells and into plasma cells, and we slowly ramp up in a logarithmic shape, and then we're going to peak maybe at 10 to the 3. And then the infection's over, we've killed off the antigen, and it quickly drops down to close to 0, but not quite as close as it was before. Now, if we have a secondary exposure, say we have a secondary exposure to varicella, this time, because we have lots and lots of memory B cells, we have a lot more B cells with this, specifically with this antigen on them, waiting along in the lymph nodes. So this time, because we have way more, it's not going to take as long for the antigen to come across the correct antibody because we have so many memory B cells. And also the memory B cells, when they differentiate, they're already starting out with a significant number, so they're going to divide and double much, much quicker. So we're not going to have much of a delay, and it's going to ramp up very, very quickly, and it's going to go much higher than it did before, and we're going to get rid of the infection likely even before we have symptoms. Okay, so that is how primary and secondary immune responses occur. So immunoglobulins. Let me go up here and get a fresh screen. Immunoglobulins are these antibodies that respond to specific antigens that are floating free in our bloodstream. And we actually have 5 different types that we've recognized, and I'm not going to go into the form of each one, but I do want to touch briefly on the function of each one. So there's IgG, there is IgM, there is IgE, there is IgD, and there's IgA. And each of these sort of they each have a slightly different shape, and they have a slightly different function. IgG is highly present and active in the serum. So it's very important for dealing with viremia and bacteremia. And IgM is actually very important for what's called host-naive viremia and bacteremia. So IgG is sort of a collection of immunoglobulins that we have been exposed to before. So it's created by plasma cells that are in the lymph nodes based on that have differentiated from B cells that have seen infections before. IgM is sort of produced randomly from B cells that have never been exposed to infection before. So these are prominent for what are called host-naive infections, particularly in bacteremia. IgE is prominent for parasitic infections. So, worms and amoebas and multicellular organisms. IgD is not really well understood at this time, and IgA is very prominent. It's actually sort of a part of the barrier system. It's very active in the skin and on the mucous membranes. Okay, and that's pretty much all I'm going to talk about with the immunoglobulins at this point.